Hello everyone, and today our guest is Dr. Krishna Bhat, Principal Scientist and Protein Science Manager in Sweden. With over a decade of experience, Dr. Bhat has contributed significantly to understanding bacterial systems and antibiotic resistance through structural biology, notably cryo-electron microscopy. So he has held key research roles at Umeå University, Thermo Fisher Scientific, and University of Graz, and earned his PhD in molecular microbiology, complemented by the masters from Uppsala University. So, a published scientist, workshop leader, and global collaborator, Dr. Bhatt is a passionate advocate for combining academic insight with industry applications. So, Dr. Bhatt, thank you very much for joining this. First of all, my question is, could you walk us through your academic and career journey from India to Sweden and from biotech student to principal scientist? What were the major turning points, please?
Yeah, hi, thanks for inviting me today. Um, I was working for, when I started my academic career, I was doing my education in BTech biotechnology in India. So I started with my bachelor in technology, not as bachelor in science, but as usual. And then from there, I moved for my master's to do in applied biotechnology at Uppsala University. From there, I learned more about proteins, what protein biochemistry is, what we can do with proteins. So when we talk about antibodies today, we talk about enzymes, we talk about proteins, we talk about a lot of different molecules that we study. They all have therapeutic importance. They all have the proteins play a huge role in our body, and that kind of caught my attention, and I was really interested in understanding how does we can engineer this protein in order to do different things. So some proteins we have in nature occurring, and some we can do recombinantly expressed in different organisms, and we can change the protein the way we want to, and that's called protein engineering. And this is what fascinates me a lot. So that's what caught my attention, and I was very interested in exploring more into this. And that's how from a student, when I was doing my bachelor's, from there I did my master's, and from my master's I got into this field, as to say.
That's nice. That's good. And also, you worked with the cutting edge tools like Cryo-EM, NMR, MELS. What are the biggest challenges in decoding protein structures, and how do these technologies help?
Yeah, so for Cryo-EM, I was working with Cryo-EM for my postdoc during my postdoc project at Umeå University. I started in 2021, and we were looking at structures, megadalton complexes, which are pretty huge in size. They form on the membranes of bacteria, and cryo-EM is a technique which, with electron microscopy, we can see the proteins with our naked eyes. So that's what is the biggest achievement that we have with cryo-EM in this space. So, um, and it is good. Cryo-EM is good for big proteins, really large proteins, megadaltons complexes. Somewhere around higher kilodaltons to megadalton complexes are what we will get in cryo-EM as a good starting point. Um, for NMR, it's the other way around. So in cryo-EM, we can see only big proteins. In NMR, we get good data for only small proteins and disordered proteins. So if you know protein biology, we have primary structure, secondary, tertiary, and quaternary structures of proteins. So in NMR, if you have no structure at all, so if you have disordered, then also we can acquire some really good results from NMR spectroscopy as well. And NMR spectroscopy is also very advanced in establishing and studying protein-protein interaction studies, or protein ligand interaction studies, and 1D and 2D spectra and 3D spectra. Using 3D spectra, we can also calculate the structure or know the structure of a particular protein as well. So these are these two different techniques. And then we have crystallography as well, which is in between both of them. And the crystallography is much more advanced in it can work for small to really big proteins. Yeah, so that's a different story. With SecMols, SecMols is mainly what we use is for understanding how polydispersed and monodispersed a particular sample is, meaning when we express the proteins tend to aggregate, meaning that they tend to stick to each other and forms higher oligomers or higher complexes, which we do not want. We want basically, if you are making a therapeutic products of a protein, we want it to be pretty monodispersed and we need to have similar kind of molecules in our sample instead of having them to be sticking together, forming higher oligomers or forming higher different kinds of oligomeric states. So we do not want that. So SecMols will give us that information as to how monodispersed your sample is, how well behaved your sample is at a given point. And that also, it's not a hydrodynamic radius. So hydrodynamic radius is when you have a molecule surrounded by a liquid. So the molecule actual weight is also governed by how it is present in a particular liquid, and that's known as hydrodynamic radius. In SecMols, it gives you absolute value of your particular size of the protein that you have in the sample. So that is a good thing about SecMols. And this is where it has in a lot of pharmaceutical industries also these days. We really want to know how monodispersed or what is the size of your protein in the sample is, which is a kind of indication of the quality of your sample. So that's where we use SecMols quite a lot as well.
So, Krishna,
Yeah.
about this protein, everything, the daily use, and also for the antibiotic resistance, you also involved in this, and also the industry and academic projects. So, in the daily life use are the success stories about the practical applications and the implementation, or you can see some practical tips for the general audience who don't know about depth of these kind of things. Just if you want to share about that.
So what so about the bacterial um, antibiotic resistance or
Yes, yes, antibiotic resistance and about the protein, the overall your area of expertise. So if you
yeah.
want to tell about there something applications where the general audience who don't know about the depth, they can
Yeah.
understand well about that.
Yeah, so protein basically is a molecule, is a macromolecule which we have in our bodies, which we have in plant, we have in any kind of food products. You take anything, any surface you take today, everything has protein. Every material has a protein, and they are all made up of protein. They play different enzymes. The different enzymes and different protein, they play different roles in the body. So our body has a lot of therapeutic relevant proteins as well. So these are molecules basically, and you have the central dogma where you have from DNA, you have RNA, from DNA to RNA, and from RNA you have something called translation, which results in protein. And by engineering the DNA, you can also engineer the protein. So people have started using this DNA constructs and the design of DNA in such a way that you get your protein which can do really different things than what it is supposed to be doing in the nature, for example. And you by using this quality of a protein, you can design any therapeutic drugs, for example, anti-cancer drugs, proteins which are involved in blood-brain barrier crossing in the brain for different kinds of half-life extension in a body. You can do several different things with this stuff. Now, recently we have had COVID. So everybody was talking about vaccines. So in vaccines, what you inject is also the RBD, receptor binding domain of spike protein. It is a protein which is present in COVID, the virus. on the
But
surface.
your specific area where you are working, can you please elaborate that specific
So
dimension
I
which?
work with all of these things. This is
Ah,
my,
okay, okay, okay. It's
I
very
work
good.
with everything that I'm talking about. I work with this thing, and I also work with vaccines. I work with making these vaccines. I work with making these proteins for injecting into the animals for generating immunogenic response. And from immunogenic response, I also make antibodies, which are again proteins. Antibodies are nothing but proteins as well. They have their functions. They bind to antigens. So this is the whole work that I
So
do.
if someone wants to pursue career in this protein science or biotech R&D, what skills or platforms do you think they should start learning? And because whether all the proteins they have to work or they have to choose one specific area, or what kind of this in a master, PhD, and postdoc, so that they can understand well, please.
It depends exactly what they want to do. Some people are interested in a very particular kind of research area, and then they want to dig deep into that particular research area, and that might have a particular proteins which are involved in the reaction cascade of a particular mechanism. So that's a very niche. That's a very, very concentrated on a one only particular subject. But if you want to work for an industry, or if you want to have your area of interest in this field, if somebody wants to have a career, I would say that people should start with molecular biology in their bachelor's and understand structural biology of proteins, and try to learn techniques on how to clone DNA, how to express this clone DNA into different expression systems, like bacterial expression systems we have. Today, we can use yeast, we can use several mammalian cells like CHO cells, HEX cells, the different mammalian cells. So try to learn all these techniques, how we can maintain these cell lines, how we can express proteins in these cell lines, all these things, and how to purify these cell lines. So purification is also one of a main, a very huge niche that we have, that people need to, if they want to work in protein biology space, these are the techniques they need to know very well, to be hired by a company.
That's good. And Krishna, about your journey, for example, what the challenges you are faced regarding your career and your, study during until postdoc. So, what were the challenges? And just for the audience who want to follow the same path, they can know well better, well in time, so that they can tackle them in a well mannered. Yes.
Challenges, it's not like software or IT, or I mean, you do not have a lot of jobs in this field. This is the one of the challenge. If you take PhD, or if you want to do research as such, then you need to have a kind of passion to become professor. You need to become a PI. You need to do principal investigation. So you need to have sort of a group, a team. You need to always write grants. You need to apply for grants. You need to get money. And that is one route people take. And that's a very usual route what people take. But I wanted to more work for an industry, and work for a company where we can look for different kinds of make products which are therapeutically relevant for people, which have therapeutics importance. And so for me, I wanted to be in this space. And this was the main challenge that I had, was that the jobs are not as many as other areas of expertise probably. So one has to be very clear about what they are looking for, and need to work towards a very specific goal, and a specific question, where they want to be. And yeah, so this
And
is
what do you think about after bachelor they should focus with some specific countries for master and then PhD and postdoc, or no?
in my field. I think people minimum have a PhD. They need to have PhD at least to get into good positions. Even in companies, or if they want to go in academic track to become a PI, or a professor. then also I would say that it would be nice for them to have a PhD.
So
In companies, people can also get jobs with masters only, but then they might be in a lower positions. Like, I mean, they might not go into decision making positions, or they might go to these positions after several years of experience.
I got your point, but some specific countries about your area of field, I mean field, there are some specific countries or industries they should target, or it is everywhere, I mean, on the globe.
I think it's everywhere. US is good. There are a lot of jobs in the US. Sweden is doing great. Sweden has always been very innovative in protein science research space, and the area where I'm located, Uppsala, Stockholm, it's a hub of a lot of purification techniques and how we analyze the protein and all that. These things, these methods have evolved in this region of Uppsala and Stockholm. So yeah, I mean, I would say any country, I do not know. I have no specific input about the country, but I know that in US it's easier to get jobs than in Scandinavia, for example. There are more jobs there, but in Scandinavia there are other perks to live in Scandinavia apart from finding a job.
So, as far as the you mentioned that IT or something, not integration of AI and machine learning, it is there, do you see where it can be applied in structural biology heading, or is there any example in your current work or
Mm.
something like that?
Yeah, so the recent Nobel Prize we got in chemistry was structural biology, AlphaFold. It's an integration of AI and machine learning in structural biology. So AI and structural biology are working very close in a close knit these days. So we have a lot of machine learning and AI algorithms which are doing a lot of things in this field of structural biology and also immunology. Like, for example, nowadays there are also a lot of companies, they are trying to generate immune response or antibodies without injecting an antigen into an animal. So they are trying to remove the animal model completely and just have just um, sequences generated out of machine learning and AI algorithms, where you can have antibodies and binders which can potentially cure a disease.
And your work also involved AI or machine learning applications or something like that?
We do, but I work more on wet lab. So I work more on making these proteins than involved in generating
So
AI.
making the protein do not need something machine learning, AI or anything kind of things. Just
No, making the protein would require one to have hands-on experience in wet lab and two
yes.
techniques to do the techniques and to do the process and the procedure protocols. This
So
is what it.
it means someone can also pursue this career where only hands-on experience is required, and the other one is machine learning or something where techniques are used. Yeah, yeah, something.
Yeah, or both can also be done. People
Okay.
can learn both as well. They can do machine learning, they can generate the sequences using machine learning, and they can make these proteins. What they have generated from machine learning, they can make these proteins in lab as well.
So you also spend time in Canada, and how do you see the time in Canada and Europe influence your mindset as a researcher?
Yeah, that's it's a very uh, I would say that Canada or Europe, they have different way of working. They have different working styles. They have different way of how they approach problems and stuff. I would say I would prefer Europe. It is more. I think Europe invests a lot in research, and and yeah, so in Europe has a lot of for a researcher, I would select Europe for example. But if somebody wants to have startup culture it's very good in the US. In Canada, it's a very it's I would say it's not as big of a market for life sciences like we have in the US, for example. So the companies, if somebody wants to work, are pretty less, but nonetheless, they are also doing good job.
So you are comparing only about your field. Otherwise, people prefer Canada over Europe. I think so.
Yeah, I'm talking about my field. When I was working there as during after my PhD, I have spent around 1 year in Edmonton. And Edmonton is in the west coast of Canada. There, for sure, the opportunities are very, very limited. There, for sure, in my field, there are hardly any companies as well. So people have to be in Toronto or Ontario, these regions, to get something in the field. Um, or people can work as a postdoc at the universities, working for long contracts and all that. So I mean, again, it's very subjective. Also, in Sweden, there are places where you do not have a lot of jobs. You have to be in Stockholm, Uppsala, or Gothenburg regions in the south to get more opportunities. It would be the same in the US as well. I think I have never been in US, so personally, I do not have any reference for US, but this is my observation about Canada and Europe.
There's and also Krishna, just a general perception about Canada and Europe, because you can well compare it, because it's rare that someone coming from Canada and come to Europe, something like that. It is special to you, and you can compare well. And the for the audience who want who are just dreaming Europe, irrespective of the field, whether the jobs are there, something like that. Can you just pros and con comparison between Europe and Canada about your observations about both environments?
Canada, I would say you don't have to, if you can speak English, that's fine. You will find jobs in Toronto, at least. I know in Toronto, you will have a lot of jobs for English speaking. In Europe, most of the places one has to learn the language for sure. Any country, you need to learn the language for getting jobs to begin with. And if somebody wants to settle in this country, then you need to learn. One needs to learn the language. So this is one thing that I would say about Europe. But then the lifestyle in Europe is much better than in Canada. I would say one they have more in Europe. People get more security for their life. The lifestyle is better. For example, medical or in general pensions or anything that we have. So that kind of security is more in Europe than in Canada. Um, and also, I think that in Europe, people need to, it depends if somebody wants to settle for a long-term plan in a European country, the way to get citizenship is longer than when compared to Canada. So that would be a kind of a con for Europe. For pros in Canada is that the settlement process is pretty straightforward if there is a job and once somebody's working there. So
So it's
yeah.
also not the 5 years in Canada to get the PR.
I am actually not sure because I never looked into that, but I know that a lot of people directly got permanent residencies from India and they have moved
Ah,
from.
yes, yes, yes. There
So
is another process. Yeah, yeah.
yeah, so that was pretty straightforward. That is not possible for Europe in any country, as far as
Yes,
I
yes.
know. Yeah.
So finally, what's before ending this session? What's your next for you in terms of research and personal journey and ambitions about you, and what do you see about your future, and also for the any advice you want to share for the young researchers, please.
Yeah, so for me, the future would be I would like to stay in this current company. We are a startup company. We started 2 years back and we are planning to expand now. So in the coming 3 to 4 years, we are planning to go public and then have products in the market. So mostly I'm planning to work in this space for a while. Alternatively, I'm also having planned to get more into marketing and sales jobs where I can get an opportunity to go and speak to people in person and try to understand the problems that they have in the real life, in the real lab, and in real projects, and to try to help them somehow, which is also of an interest for me. So this is something that I'm looking for. I want to be more on a field like field application scientist or some kind of positions like that in a bigger organization is what my future plan would be. And people who want to young researchers who want to do something in this field, my advice would be to know what they really want to do from bachelor's already. If they have a clarity on where they want to go and just be focused on that goal, that would be very helpful. And I do not know probably if we are talking about in India. When I was doing my bachelor's, we didn't have any subject called structural biology during my bachelor's, but in Europe, they already have in bachelor's courses like molecular cloning and structural biology courses, which gives more input. So I would encourage everybody who is doing bachelor's in India or any other South Asian countries to take additional interest in these protein science areas and to get already get into that before they start their masters, where they will have more information about these subjects.
So thank you, Dr. Krishna Bhat. Thank you for sharing your insights and definitely your dedication to solve products, real-time world problems in the form of products is really inspiring. And for our audience, if today's session sparked curiosity or ambition, so make sure to follow Dr. Bhatt and explore opportunities in structural biology and biotech. And don't forget to share this webinar with aspiring scientists. See you in the next session. Thank you and bye bye.
Yeah, bye bye.